lecture was to just kind of go over Genetics 101. And some of the clinical consultations that I had with doctors, I had a couple comments or desires to just sort of have a refresher course. Some people, it had been a while since they had genetics, you know, in medical school. And then also in medical school, a lot of the genetics it was medical genetics. And talking about rare genetic disorders was the focus. And some people just wanted kind of just a review of basic genetics so they could understand things like epigenetics. This is like kind of one of those new buzzwords. I don't know if you guys have been hearing epigenetics a lot, but it wasn't really talked about or taught in school because it is kind of newer. So if you're like me, who went to school like 10 years ago or more, then I never was told anything about epigenetics. So I teach a basic genetic course to undergrads at NUNM, and so this is going to be just kind of a quick overview so that I'm hopefully not boring people, but at least getting you this information fresh in your mind so that when we do talk about certain things in the upcoming lectures, you guys will be like, oh yeah, I know exactly what she's talking about. So that is my learning objectives. I want to go over kind of genetics, the history of genetics. I want to talk about what genetics is now, what we're doing with it now, and kind of review some of the fundamental ideas in genetics, structure of DNA, RNA, and medical terminology or genetic terminology, because that I find is like one of the biggest questions I get from practitioners. They'll be like, wait, what does that word mean again? So, and we're going to talk about other things like transcription, translation, mutations, epigenetics, and basically anything in between, just to kind of get you guys all on the same page. So, genetics in action. I love that's my life. You know, this is the job, genetics in action. Why do I love genetics? Because honestly, I love the idea that you could take your DNA, mash it together with another person's DNA, and create life. It's amazing. It's the fundamental principle. It's what builds humans into who they are. It's what makes us different from each other, and it's what keeps us similar at the same time. It has everything to do with evolution, medicine. It touches on all walks, I believe, of science in the health field. So genes, what are genes? Why are they so important to us as healthcare professionals? Is because they really have that effect on what makes a person and then what makes us an individual at the same time. And they are the root of many diseases. When I learned in medical school genetics, it was mainly rare genetic disorders. But as everybody here knows, now that we're getting into things that more like SNP testing, you're going to see that we're talking about genetics that increase susceptibility to very common diseases like diabetes. So the history of genetics. Does anybody remember who this guy is or ever Norman Borlaug? I don't know if you guys, he was huge. He actually used selective breeding to make plants produce more and to increase their yield so that they could feed a lot of like starving nations. And this is one of the prime uses of genetics just back 100 years ago. It's just in selective breedings to increase, crop production. And then we jump into genetically modified foods, which has a huge basis in genetics because you're actually inserting a gene into a plant to change the genome of the plant. Genetics is used in a lot of different, it's not just medicine, a lot of biotechnology. But in regards to medicine, it's used a lot in creating medicine. I don't know if you guys know how insulin is made, but you can actually take the gene, the instructions to make insulin, insert it into a bacteria, and then that bacteria will create that medicine for you. While you sit back with your feet up. It's amazing that all the uses for genetics. And genes are important in development. This is one of the that term epigenetics, this is one of the main uses the body has for epigenetics. It turns genes on and off depending on when you need them. So when you're a fetus developing in your womb, in your mother's womb, you're going to be using different genes than you are as an adult. And that is the main use for epigenetics. So the history of genetics, I really think of genetics as just being around in the last, couple hundred years when we started in the biotechnology, when we started to have lab equipment that could test for it, and that's when I thought genetics originated. But it actually has been around, and the principles of genetics have been in use for tens of thousands of years. They used it for selective breeding, to domesticate animals. And noticing patterns of inheritance, that's been around for a really long time. In ancient Jewish writings, there was proof that they acknowledged the inheritance of hemophilia. They said that if a woman's son dies from excessive bleeding during circumcision, they knew that all her future sons were to never be circumcised, and her sister's sons to never be circumcised. So those patterns of inheritance have been noticed and observed and utilized for centuries. So does anybody know who this is? Is this guy ring a bell? I have one smile. Gregor Mendel, he's my celebrity crush. He's amazing. He is kind of considered the father of genetics. He was raised, his parents were farmers. He came from a humble background. He was a monk. He wasn't raised with this knowledge. He took it on himself to learn science, and he was one of the first people to really publish a clear pattern of inheritance. He's the reason we have Punnett squares, which I absolutely love as well. He taught science at a university, and he's the one that used the pea plants and the experiments with the pea plants to show clear patterns of inheritance. And he started that whole ball rolling. Why was he so successful? He was very methodical and very consistent about his experiments. So he also really helped to develop a good structure for performing experiments. He's huge. He's a huge person in the scientific history. And he, of course, like many of the major players in scientific history, died without really being recognized for his contributions. But I just assumed that he was a humble man and wouldn't have minded anyways. That's me giving him a personality. So genetics today. What are we doing today with genetics? Well, the human genome was first completely mapped out in 2003, which was a huge step. Now we're to the point where a person can send in a swab to a lab and get a huge amount of information about their genetic data. And it's not, tens of thousands of dollars to do that. And it doesn't take months to sequence. It can happen in a matter of a day. So it's been a very fast progression. And that's left us as the healthcare community wondering kind of how we utilize this tool to best, to be the best doctors we possibly can. Because I don't know if you guys are thinking this way, but in my opinion, this is really the wave of the future. I don't know if anybody's ever seen the movie Gattaca, but I actually assign that to my students. It's extra credit, but I make them watch the movie because most of them are in their 20s and have never heard of it. But I think it's an amazing idea to think that genetic information can be so readily available to people, and that we can have these master databases that can search through and look for every single different type of allele, put this picture together, and then say, you are going to die of a heart attack at age 37, like they, whatever I think it was that they predicted in that movie. So it's really that wave of the future, and we're kind of on the forefront here, doing our best to keep up with the ever-increasing amount of information, which Dr. Knapp will talk about in her research section. So let's define a genome. I just really like to kind of define some terms. I think this is almost the most valuable part of the lecture. This is the complete set of genetic information in an organism. And I think one of the biggest things that people get a little confused on is that this is all your code. This is your entire genetic code. But if anybody could guess, like if you were to guess what percentage of that genetic code do you actually use, what percentage of your genome actually encodes for genes, does anybody have a guess?
Less than 5 or less than 1%.
Yeah, it's like 2 to 3%. Exactly. Most of my students will guess something like 30 or 40%, and then they're shocked to realize how little it is. So we use little, but we use it well, I would say. And it's your DNA. and every organism has a genome. So here's some other common terms that I really like to kind of point out, and I know that you guys know most of these already, but a gene is just a section of DNA that is actually encoding for RNA, which most likely becomes a protein. Allele, this is one of the most important ones, and this is the one I love the most. You can have a gene for eye color, and then everybody has their own version of it, and that's an allele. So I have the brown-eyed allele, my husband has the blue-eyed allele, and so on down the line. So locus is a specific area or point on the chromosome. Your genotype and phenotype, that's another distinction I really like to make. The genotype is your actual genetic makeup. So I have one brown-eyed allele and one blue-eyed allele. But what you see when you look at me is my phenotype, and that's the blue-eyed, or the brown-eyed. I can remember what color eyes I have. I promise. Then you have homozygote and a heterozygote. And homozygote is just somebody who has two copies of the same allele, and hetero is one that has more variety, has one copy of one allele and a different copy of a different allele. And then this is just your chromosomes. You have the 23andMe, that sort of thing. You have X chromosome and Y chromosome if you're a male, and XX if you're a female. You have two pairs, one from your mom and one from your father. And then this graph I think is really important because I just like to show how everything moves from one step all the way, so from the DNA to the protein. I like to kind of review that really quickly. Because you have your DNA, and your DNA is your storage, your blueprints. And if I use a factory as an example, because that's really what a cell is, it's a little factory. These are the blueprints. They're sitting in storage in some room in a filing cabinet. When you want to make a shirt and you want to use these blueprints, what you do is you pull it out and you make a photocopy of it. That photocopy, that's the RNA. Because the RNA can travel down to the actual warehouse where the shirt is being made. This information on the RNA, it travels down to the ribosomes and it is turned into a protein, and the ribosomes are like the workers, the transfer RNA and the ribosomes, and then they make that final product. This is another thing that I like to point out with the gene structure. So this is a gene. It's just that one little spot in the DNA, but what's important about it, and I think I really want to point out here, is that you have this area that actually encodes for the RNA. So this is the information, but you have a whole section in front of it. And that's what a transcription factor can bind to. And transcription factors are incredibly important because these turn up or decrease the rate of transcription. So when we're talking about, say for instance, if you have a genetic defect in a gene that makes a transcription factor, and it hurts that product, so that transcription factor doesn't work very well, that is going to have a much larger consequence for your patient because that is going to damage your ability to transcribe several genes. Whereas if you have a defect in a structural gene, you're just hurting that one product that it encodes for. And I think that's a very important thing to remember when you're talking about what gene. When you're looking at, okay, so I have a mutation in this gene, and you're looking at what it does, if you hear that the mutation is in a gene that encodes for a transcription factor, I want you guys to be like, ooh, that doesn't sound good at all. Just right off the bat.
Like a photocopy, I mean, it's a function.
Yeah. So there's two kind of genes. There's a gene that encodes for like a structural protein, like a receptor or an enzyme. And then there's genes that encode for transcription factors, which then go and bind here, and they can increase the rate of transcription of several genes.
Are these transcription factors always gene-specific?
Yeah.
Yes.
Yeah, the transcription factors are gene-specific, meaning they have certain genes they affect the transcription of. And so that's the take-home message of this slide. So structure and function of proteins, this is the end product. Like what are we trying to do? And this is one thing that I love teaching students about because they hear the term amino acid all the time, but they don't really understand that proteins are just a string of amino acids. And that is so important too, because when we're talking about SNP testing, and I'll go into like evolution and why SNPs, mutations and changes in one base pairs, they tend to be more what causes alleles and genetic variation as opposed to what causes death. So a lot of what we test for is SNPs, and what is that? What effect does a single change in a base pair have? Oftentimes, what the effect is it just changes one amino acid in that string of amino acids that make up a protein. So it can have a very subtle effect or no effect or a large effect depending on how it interacts or how it changes the interaction of all the amino acids in that chain with each other and how it affects the folding. So these amino acids are joined together in a polypeptide chain. And you can see here, this is the structure of protein right here, protein structures. You come down to the primary structure, which is just that chain of amino acids linked together. Then they interact with each other based on what amino acids are put next to each other to cause a secondary structure to fold up on themselves. And then they fold up again based on their interactions into a tertiary structure. And then sometimes you can have multiple proteins come together to form a larger structure. All based on their attraction, all based on the chemistry of the amino acids. So think about it, if you just change one of these amino acids, start thinking about that, how that's going to affect the protein. It may not cause the protein to become completely ineffectual. It may just decrease its function, and we've seen that a lot, decreased enzyme function. So here, this is really kind of an overview of translation. You have your ribosomal RNAs come together in that little, I call it a hamburger bun. So you have your messenger RNA that's running through the ribosome here, ribosome there. And then you have your little workers, which are the transfer RNAs. These are the ones doing the job. And they come up and they sit next to each other, and when they do, these amino acids come together and form that chain. And as it moves through this ribosome, the chain grows longer and longer and longer until it's terminated, the chain breaks off, and then folds into that structure. That is the protein. So translation in antibiotics, just a little pearl to take home from all of this. This is how a lot of antibiotics work, is by disrupting translation. Because the ribosomal units in bacteria are different than eukaryotes, a lot of antibiotics target that exact process, and they make it so their ribosomes don't bind as well, or they disrupt the function of them. And that's actually how they kill the bacteria, is by making it so the bacteria cannot produce any protein products. So, if anybody's ever seen one of those t-shirts that says like, it has like a chimpanzee and it's like 98% difference or something, that's talking about the fact that our DNA and the DNA of a chimpanzee differs by like 2%. So really when I heard that, I was like, oh, that 2%, that must be some really important stuff. But what they found is actually it's not so much that our DNA is so different, it's that we use those transcription factors differently. We have more transcription factors in a certain area than they do, which then goes and affects the production or the transcription of a bunch of different genes. And that's why those transcription factors are so important. And that's what makes us so phenotypically different than a chimpanzee. So it's interesting, the inner working. So levels of gene regulation. That's what a transcription factor does. It regulates how much a gene is working, is utilized. So in other words, if you don't like that blueprint, you can store it in a locked box. You can lock the box that it's in and nobody can get to it, and nobody's ever gonna make a photocopy of it or make that shirt. That happens, that's epigenetics. That's how you turn a gene, you can turn a gene off. Vice versa, you can turn a gene on. So you have levels of regulation where you can either make it so the DNA is really compact and you can't access it, or you can turn it on. You can make a big messenger RNA and then cut it up in a certain way so that it either is not working, not translatable, or it's processed into a specific product that is different than if you had processed it a different way. And then you can actually make the protein, and then your body can come along and just inactivate the protein if it doesn't want it. So your body has a lot of options. That's what I want you to take home from this slide. Your body has a lot of options. Like, just because you have a gene doesn't mean you're using it. And maybe you want to use it. And you want to increase the use of it. And that, again, is where epigenetics comes into play. So DNA binding proteins. Much of gene regulation in bacteria and eukaryotes are accomplished by these proteins that bind the DNA section or sequence where I was showing you right before it, and they can increase or affect the expression of that gene. And this is just I know these pictures kind of look crazy, so I'm just going to try and sort of like show you the main point of it. These little areas before the gene, this is an area before the gene, they attract those transcription factors to bind there. And when the transcription factors bind, they increase the binding of the actual enzyme that does the transcription, RNA polymerase.
So translation is more DNA than transcription factors in RNA.
Yeah, transcription is going to be, when you turn DNA into RNA, and translation is when you turn RNA into proteins. Did that answer your question? Great. An example of this is a lac operon. Now I'm just jumping into some serious genetics here, which I absolutely love. This is an example of why the body needs to be able to turn genes on and off at the flip of a switch. It's because your environment is constantly changing. So in the example of these bacteria, if there's lactose, that sugar lactose in their environment, they want to be able to gobble it all up. But the truth of the matter is that it isn't always there. It's usually glucose. So why would you leave the genes on that help you break down lactose? Why would you leave them on all the time? That would exhaust your resources. So the body, the cell, the bacteria, has the ability to turn those genes on only when there's lactose present. And the lactose can actually go in, and through this series of very confusing this series of steps that I make my students memorize, but I'm not going to overwhelm you guys with, it can actually go, the lactose sugar can be broken down into something that actually binds a transcription factor. This little thing here, this comes from that sugar, lactose. It binds this transcription factor, which can no longer block the transcription of the gene. The gene gets turned on, and now you have enzymes to break down lactose. And the take-home message of that is how diverse our system is. It can really respond to our environment. Our genetics can respond to our environment. And I think that's a really powerful thing to take home. And that's epigenetics. And that is, we're not going to, I'm not going to jump into it, but I just really I see sometimes there's a little confusion on what epigenetics is. And I'm going to go over. Basically, epigenetics, your body can control transcription of a gene in 3 main ways. It can actually methylate the DNA, which I think a lot of people have heard that one. It can modify the histone that holds onto the DNA, and then it can actually, RNA can actually affect. And I'm going to show that really quickly. So here's histones. DNA is wrapped around histones because it is like, if it wasn't, it would look like spaghetti all over the place, just craziness. So we have so much DNA, we have to have an organization system, and that system is to wrap the DNA around a histone. If that DNA is wrapped tightly around that histone, then you can't get into it to transcribe it. You have to loosen it up first. And that's a choice the body has. And when it makes that choice, that's an example of epigenetics. So that's one main example. DNA methylation. What does that do? If you stick a methyl group on DNA, it's like a roadblock. You can't, the RNA polymerase can't land. It's like basically taking a big truck and sticking it on a landing strip. DNA can't, the RNA polymerase can't land, and then it cannot transcribe that gene. And so the gene is turned off. Then you can actually, has anybody ever heard of X inactivation? This is the coolest thing ever. I'm about to explain calico cats to you. So every woman has 2 X chromosomes. Guess what? We don't need 2. So we only use 1. What do we do with the other one? We turn it off. We do it using RNA. The gene, the X chromosome, will encode, the X chromosome that gets turned off will encode an RNA that then coats it and turns it off. It actually turns itself off. It has a little gene here that makes a long string of RNA called xRNA, which then goes and coats that X chromosome. And once it's coated, then other enzymes come along and actually methylate it and turn the whole thing off. What's fascinating is that this happens different, your body chooses which X chromosome to turn off, and it's different for every section. Yeah, so
For every cell, it is different.
So when you see a calico cat, has anybody ever been told that calico cats are only females? It's because their spotted coat, you can see like the different colors throughout their coat. That is because in a certain color, 1 X chromosome is turned off and the coat color is on that. So if you turn the orange X chromosome off, it'll be a patch of dark black. But just over a little bit, maybe the other chromosome was turned off, so now it's orange. And that modeling pattern shows you how varied it can be in which X chromosome you decide to turn off. And that's an example of epigenetics. This slide is meant to show you all the different types of mutations. We're jumping into mutations now, because that's really why you guys are here, to talk about mutations. There are a variety of different types of mutations that can happen. You can have a whole section of a gene be removed. You can have it be picked up and then put over into another part of your chromosome. You can have an entire leg of a chromosome come off and go and insert itself onto another chromosome. There are so many different ways that you can mutate DNA if you so choose. One of the ones that we talk about the most is a base substitution. And that's when you're just changing one tiny base pair in the DNA. And what's that going to do? That's a single nucleotide polymorphism. That's the SNPs that we're talking about. And why is that so important? Because genetic variation is important for evolution. And genetic variation is going to happen most often due to a SNP, because if you just change one little part of the protein, it's most likely to still be functioning. These little SNPs are not often going to kill an organism. They're going to slightly change it and give it genetic variation. And what's the importance of genetic variation? It's evolution. If we don't have genetic variation, if we all look the same, then one disease could wipe out an entire species. This is the whole reason why we're able to survive. And there's actual proof that when you're in an environment that stresses you out, your mutation rates increase, because the evolution says, okay, this is a stressful environment, something's going wrong. If I mutate my species and create genetic variation, there's a chance one of them's going to survive to reproduce. So if you think about mutations as an actual purposeful thing that we've been selected for to do, you can kind of see why, why there's some importance there. But what happens with us, what we're seeing most often is it is creating disease. And so that's why it's clinically relevant to us. And we do have, I just want to touch on it, we do have repair systems built in to actually go back through and repair damaged DNA and mutations. What happens, why do we see these mutations then affecting us in our health? It's when our repair abilities have become overwhelmed. and we're not able to keep up with it. So if there's like a little damaged part of the DNA, we actually have enzymes that will come in, cut that damaged part out, and replace it with a brand new piece. If we have too much environmental toxins or exposures, those systems become overwhelmed. And finally, single nucleotide polymorphisms. These are a site where the genomes can vary wildly, and that's why they're important. And this is why we have this whole new field of genetic testing that is starting to focus on SNP testing.